Okay, so we're going to begin the membrane structure and cell membrane transport. So the cell membrane, we want to think about the structure of the membrane. We talked about the phospholipids, which are the main components of cell membranes. And so we're going to carry on that discussion here and talk about what are some of the properties that the various structures offer to the membrane. So our learning objectives to describe the structure and major function of phospholipids and the plasma membrane, to explain the role of chemical, electrical, and then the combined electrochemical driving force in the passive transport of substances across a membrane, and distinguish between passive transport and active transport. So again, a lot of the reason that we want to talk about the membrane and that we're so interested in the membrane is because this is the basis for transport. Based upon the structure of the membrane, that will determine what things can go through and what things cannot. So let's start with that first objective. And now we usually describe the structure of the plasma membrane as the fluid mosaic model. So I'm going to start by writing that up here, the fluid mosaic model. And what does that mean? This pretty much means that the cell membrane is not a rigid, fixed structure. There's a lot of flexibility, there's a lot of movement, there's a lot of fluidity in the membrane. That's what the fluid part means. The mosaic means that there are many different types of structures. So it's really a collective membrane that's formed by different types of proteins, different types of lipids, different types of carbohydrates. And so we have cholesterol, so different types of fats. So basically, it's really a mosaic of several types of structures that give the collective properties to the membrane. So let's begin to draw that membrane here out. So we have our phospholipids, which we know have the polar head and the non-polar tails. And it's really this bilayer that is facing where the fatty acid tails are facing the center. So let's kind of draw this out first. We're going to leave some gaps here for some of the proteins. I know these look like terrible stickmen, but they're not. So let's do the two sides of the membrane. So remember, it's two leaflets that are facing each other. And again, I'm going to fill in some of these larger structures here so that we have some examples of some proteins. We'll talk about what this is. But this comes right through. Oops. Let's do that again. This comes right through the center of both leaflets of the membrane. Okay. And then we have some other proteins that only go through parts of the membrane. So we'll draw some of those in. So let's keep drawing in our phospholipid bilayer. We're going to add in some of the cholesterol rings. These are another feature that we find in the membrane. And then lastly, I'm going to put some of our carbs that stick off of the outer surface up here. And we'll go back and describe what these things are. So the first thing we want to think about is what are the two faces of the membrane? So here is going to be the extracellular fluid out here. And then down here is going to be the intracellular fluid. So this is going to be on the inner surface of the cell, right? Essentially the cytoplasm. And then if we think about it, we want to describe the aqueous environment. So the aqueous environments are going to be on the inner surface, right? And that means that this surface is polar. This is also an aqueous surface. So lots of water or fluid, also polar. And then on the in between of our membrane, so this inner surface here, this is going to be non-aqueous or non-polar. Oops. Non-aqueous or non-polar. And we discussed this already, the fact that non-polar molecules cannot exist in the center of the phospholipid bilayer, right? In order to pass through the membrane, it cannot be a non-polar. It has to be a non-polar molecule to exist in this center, excuse me. So let's describe the phospholipids first, right? This is called the phospholipid bilayer. And what are some of the properties of the phospholipids themselves? First of all, they are amphipathic. Amphipathic. And this means that they can exist in, span across aqueous environments. They have the non-polar fatty acid tails and then the polar head. So they can span across both aqueous and non-aqueous environments. So they're perfect to exist as the separation between the intracellular and extracellular space. Another feature that they offer the membrane is fluidity and flexibility. All right, so fluidity and flexibility, which means that they allow some things to go through, others to not, but they also give some movement to the membrane. They're not rigid or fixed structures. And we'll talk about some of the cholesterol function, which is to break up the interactions between these phospholipids. Another feature that we want to say here is that they are impermeable to water. Impermeable to water, okay? And polar molecules. They're impermeable to water and polar molecules. In other words, a polar molecule cannot exist in the center, so it cannot cross the entire membrane. Okay, let's talk about our proteins. Now, proteins can act as several different structures in the membrane. They can be receptors, they can be transporters. They can also act as signaling molecules, which we'll talk about, which is one of the phospho or glycolipids. When we think about proteins, they can be transmembrane. So this protein here that goes right across both leaflets is called a transmembrane protein. Transmembrane protein, all right? And so we want to describe transmembrane proteins as being amphipathic, right? They also have to be amphipathic in order to exist on both surfaces of the membrane, both the polar ends and the non-polar center. So they have to be amphipathic to span the entire membrane. Okay. We also want to think about the fact that they are permanently fixed into the membrane. They are permanent in the membrane. So unless the membrane dies altogether, unless the cell dies or the membrane is damaged, Integral proteins are permanently fixed, right? So they're fixed. Let's now talk about peripheral proteins. And peripheral proteins are usually on the outer leaflet. So usually on the cytosolic side, did I say outer leaflet? I meant the cytosolic side of the cell membrane. So peripheral proteins, they are found on the cytosolic side of the membrane. These are easily movable, so these can easily dissociate. And dissociate means that they can move away from the membrane, float across the membrane, right? Not across as in spanning the membrane, but they can move around on the cytosolic surface. So they can move position on the inner surface of the membrane. So they can easily dissociate and move around. They're going to be anchored to the membrane by cytoskeletal elements. Okay. Actually want to clarify here. So when we think about transmembrane proteins, they can span the entire membrane. They can also exist to span just one side. So I want to clarify that these are also transmembrane. And I'm going to add that down here. So they can also span one side only. So a transmembrane protein goes through the membrane, whether it's the full length of both leaflets or just one side. Peripheral proteins are always on the cytosolic surface, so they're always on the inner part of the membrane. They do not go through the membrane. Let's look at cholesterol. And cholesterol also helps to offer fluidity and flexibility. These are these fatty rings that are inserted into the membrane. So these increase the fluidity and the flexibility. Okay. Another thing that cholesterol does is it breaks up the phospholipid interaction. So it prevents the membrane from becoming crystallized. And we say that because it spans in between some of the phospholipids and prevent them from rigidly joining to each other. So it will decrease the rigidity. Oops. Decrease the rigidity and prevent crystallization. Okay. Cholesterol also prevents membrane leakage. So because cholesterol is a fat, it just helps to reinforce the non-polar movement of molecules. So it's basically going to prevent any leakage of water through the membrane. And that is because it's a fat, so it is impermeable to water. Okay, so that's your cholesterol. Another structure we want to think about in the membrane are the carbohydrates. And I've put the carbohydrates up here in red, but let's describe their functional properties in the membrane. So carbohydrates. And carbohydrates are important for a couple things. So they're going to extend on the extracellular surface or extracellular leaflet of the membrane. not necessarily the intracellular leaflet. And the reason for that is because they help with cell identification, cell tagging, and cell signaling. So cell identification, tagging, and signaling, okay? And that basically means they help one cell identify itself to others and even send signals or messages from one cell to the other about things that may be going on in the cell or any requirements that the cell may have, right? So that cross-cell signaling and basically cell tagging. They are going to be covalently bound to lipids or proteins. So covalently bound to lipids or to proteins. So if we have a carbohydrate or sugar that is bound to a protein, right, here's one of our proteins, that's going to be called a, where are we? Okay, yeah, that's going to be called a glycoprotein. Okay, so a glycoprotein is a sugar covalently bound to a transmembrane protein. And then a glycolipid is a sugar bound to a phospholipid. So depending on what type of membrane structure, or sorry. Okay, so that's correct, a glycolipid. Okay, so glycoproteins and glycolipids. All right. So this is basically a model of what's happening with the cell membrane that describes all the different players, all the different components, and the functional properties that they bring to the membrane. How do they help the membrane function? How do they increase the fluidity, the flexibility, span the membrane? So the transmembrane proteins, for example, we'll talk about some structures that these can be, but transporters is an example of this, and we'll talk about that coming up. So I'm going to put example here. These can be transporters. Okay. The peripheral proteins, these can be receptors, right? So we'll talk about some different examples, but essentially all of these structures come together and are very important in the overall function of the membrane.